Hi everyone, welcome to my channel Srinivasa Biology Tutorials. For this session to discuss NCERT Class 12 Chapter 4 Principles of Inheritance and Variations, Linkage and Recombinations. What is the linkage? What is the recombination? See, Morgan carried in the several the dihybrid cross in the Drosophila to study the genes where the sex linked. See, here Morgan did as in that experiments on the Drosophila. Here that entered the genes are located on where? In that in a X chromosome. They present as where? In a X chromosome. Okay, and here the cross is similar. The dihybrid cross is carried by as in a Mendel pre-plant. So Mendel, how did the experiment the dihybrid cross? Here also the Morgan also did as in the same experiments. The Morgan hybridized as in a, he took as in a few characters. He took as in a few characters, that is in a yellow body, that is in a that yellow body, and then in a white eyes. This is what is a female. It is what? Female. And next one is that is brown body, and then red eyes. Is what? It is in a male fruit fly. So he did as in a cross. See, in these the intercrossed there the F1 progeny. He observed the two genes. They are segregated independently of each other in the F2 generation, getting as in a 9:3:3:1 generation. That is the phenotypic ratio is get like as in a Mendelian. The accepted as in that the two genes of independent. So that the two genes how to be what you get as in a the dihybrid cross phenotypic ratio you also get as in that same process. Okay, and after Morgan and his group, the new genes, they are located as the X chromosome. They saw that quickly when the two genes or dihybrid cross with a shoot up with the same chromosome. So the chromosomes as present here, the chromosomes are present on the same chromosome. Here the proposition there parental gene combined with as any they are higher than non-type. In the Morgan attributed as in the physical association with as any two genes is called as a linkage. So here when you take as in the two chromosomes, they are completely the physical association. Here the physical association of the two genes here is called as what? Linkage. And here after that describe the physical association of the genes or genes on a chromosome is called as a recombination. So in this complete describe as in the two physical association of the gene of the chromosomes is called as what? Recombination. Here physical association, here gene of the chromosome they are completely they get fused to form as what? Recombination. Yes, okay. Next, describe the generation of the non-parental in combination. So in that generation to generation to get forms as what? In a recombinations will becomes and non-recombinants also comes in generation to as any next to generation. And Morgan and his group also found even the genes were grouped on the same chromosome. So here Morgan also they explain as what? The entire the chromosomes are to get moved from as generation to as any generation. In some genes are very tightly linked, it shows as in a low recombinations. They are given as in a cross A in that map, and while the loosely the linked their recombinations, it has for the example the found the genes where the yellow, white and yellow is very tightly linked. It is show as in a 1.3% of recombinants, and while white and miniature they show as in a 37.2. That is in a that non-recombinants we get showing. The recombinations they get show. Okay, and see here I'll tell you what is that ratios. I'll explain the diagram. Okay, next, and after his students means Morgan students Alfred Stutwant, he used as a frequently the recombinations between the gene space, the same chromosome, the measure the distance between the genes mapped there in a position of the chromosome. Today we are genetic mapping. We are seeing that in a genetic mapping. So the scientist was whom? Albert Stutwant. He was explained about as in a this mapping. Okay, it is extensively the starting the point the sequence of work, the genomic done the cases of any human genome sequence, the project described in later. So in this how do they get forming, he was clearly explained here only. Okay, let us see here. Let us see, it is a cross A, it is a cross B. So here when the genes are, when see here genes are there completely closely they are present, then what happen? Here get as any their parental The parents visit comes as a more and recombinants comes as in only less, that is a only 1.3%. And here recombinants comes as how much? Here 37.2% the recombinants has comes. Okay, now here loosely packed, here they are loosely packed, here get as in tightly packed, so parental comes more. Okay, here recombinants comes as any less, but here comes as any more. With this any complete comparison, it is in a completely yellow white, it is in a completely white and miniature. Okay, this is in a completely the mapping of any what at a process of alphabet sequence what they are explained. Okay, next what is polygenic inheritance? It is polygenic inheritance. See, Mendel studied mainly that describe those either traits as in distinguish their alternative forms such as in a flower color which either to purple or white. But you look around as in a wild find that any many traits which are not shown as a distinct trait, the occurrence of the niche they are spread across gradient. For example, you have seen that in a human does not just having a tall short peoples to distinct alternatives but whole range of the possible heights. So when you observe the different peoples, they are short and long, the different variation they show depends upon the gene variations. Okay, such traits are generally the controlled by as in a three or more genes or called as in a polygenic inheritance. Why they are forms like means the more than one or three genes they are complete they are affected, so that purpose that character they are expressed, that is called as a polygenic. Poly means what? Many. Genes means what? Genes. So more than two or three genes they are affected, that is called as a polygenic inheritance. Based on the involvement of the multiple genes, the polygenic inheritance takes in the account of influenced environment. So it is mostly takes place as what? Influenced environment will be that takes place. Okay, next the human skin color, another the classic example for this one. So in this complete polygenic inheritance, another example is what? That is in a skin color. That is what? That is in a skin color. Skin color. Next, a polygenic traits of phenotype that reflect the contribution of the each allele, the effect of each allele attribute. They understand this better and assume that in a three genes, that is in a gene, B gene, and three genes, they are controlled by as in a skin color of the human, the dominant, that is in a ABC. The responsible for as in a dark skin color, and the recessive, the small a, small b, and small c in a light skin color. So here their capital, they completely capital A, capital B, capital B responsible for as in a their skin color, correct? That is in a, and the recessive is what? Small a, small b, and then small c. These are completely recessive parents. So they are completely controlled by as in a that skin color also. Okay, and the genotype of the alleles, the dominant alleles is what? Capital A, capital A, capital B, and capital C, capital C. And while they have a dark skin color, that will be their recessive parent alleles is small a, small b, and then small c, small c. That is in the recessive parents. Will have in a the light skin color as an experimented the genotypic which has in three dominant alleles. The three recessive alleles we have in a intermediate skin colors we are noticed. So when you cross, what happened? It cross as in a they're completely their capital A, small a, capital B, small b, and capital small c. This the heterozygous characters we are noticed easily. Okay, and after in this, see in this the manner of number of each and type of the alleles, the genotype would be determined. That is in a complete darkness, lightness of the skin color they are individual. So this totally their genes are there affected by as in a in that in a phenotype or in a genotype. Okay, it is completely called in a completely polygenic inheritance. Okay, and then what is that pleiotropy? Pleiotropy. See, pleiotropy. You have so far seen the effect of the gene, the single phenotype or trait. These are, however, the instant we have in a single gene can be exhibited as in a multiple phenotypic expression. So they are showing as in a single gene, but they show as in a different variations. They show as in a the different variations. In multiple variations they get show in that in a process. That is completely process is called as what? That is in a process of in a pleiotropy. And the genes are called as pleiotropic genes. The underlining the mechanism, the pleiotropy, most cases the affect the gene, the metabolic pathway, it is contributed as in different phenotypes. So the different phenotypes they can completely clearly they're showing as in this process. Next, an example of this disease, the phenylketonuria. So in this completely pleiotropic example, that is in a phenylketonuria, which occurs to the humans. The disease the caused as in the mutation genes the code of the enzyme phenylalanine their hydrolase. It is in a single gene mutation. It is in a phenylalanine hydroxylase. It is in a single gene mutation. It is takes place as in the only for the pleiotropic gene, and it is manifests itself as a through as in a phenotypic expression characters. The Mendel their completely retardation and the reduction of in a hair and skin pigmentation. Here, hair and pigment completely skin pigmentation takes place with the causes as in a pleiotropic gene. Okay, this is next one is sexual determination. Next one is what? Sexual determination. The mechanism of sexual determination has always have been the fossil before the in genetics. See, the initial curve there about the genetic and chromosomal mechanism, the sex determination can be there traced back in some experiments carried out in the insects. Fact there in cytological observations made of the number of insects lead to as in development of the concept the genetic chromosomal basis of the sex determination. So here what happened, the different chromosomal mechanism they show as in the different expressions that depending upon the genes how do the chromosome their behavior takes place, the automatically they show as in their completely their characters. Okay, that is. And hanging in 1891 he could as in trace the specific nuclear structure and through as in the spermatogenesis and few insects, and it is also the observed as in him in 50% of in the sperms received the structure of the spermatogenesis. So the scientist was in a hanging, he first there noticed as in the collection of in a some completely there some specific nucleus in that in a spermatogenesis after he did as in experiments, whereas in the 50% of the sperms he did not receive it. So 50% only there he received, 50% he not completely the received. The hanging give as a name that structure of in X bodies. What the 50% did not they receive, and which if the 50% spermatogenesis is received, that is called as what? That is in X bodies, but could not the explain the significance further. The investigation of that in a scientist lead to the in a conclusion that in X bodies hanging was in a fact in the chromosome. So he not completely explain about as in a what is that X bodies, what is X bodies, he not completely they get explained. And that why is given the name of the X chromosome. Why they given as an X chromosome means, see here they show as an X bodies, it is an X chromosome, it is in a Y chromosome. Okay, here the mostly the females comes as in what, it is in a completely XX chromosome. Like when you observe as in a, it is in a completely male Drosophila, it is in a female Drosophila. See here also the X chromosome, Y chromosome, and here completely has an X chromosome that in a female Drosophila. Okay, and here it next he was also is observed that in a large number of insects organisms the sex determination it is an XO type. The mostly they having as what, XO type. Here the all egg bear as in their additional of X chromosome based on their other chromosomes, they are completely called as in a autosome. So when you observe to as in the XX chromosomes, here they show as in what, complete XO type is common, and he was they say about as in a the other X chromosome based on a other chromosome is called as what, they are the completely autosomes. They completely autosomes, and some of the sperms bear X chromosome, whereas some does not there appear. So some chromosomes there appear and some chromosome does not there appear. Already we discussed that one only. Okay, and the egg fertilized sperm having as X chromosome become as in a females, and the fertilized sperm do not have an X chromosome, that is in a male. So there's some fertilized the sperms do not have an X chromosome become as what, males. And do you think the member of chromosomes in that in a male and female the equal? So they having about the completely X chromosome is present in a female, XY is present on that in a male chromosome. Okay, the X chromosome determination of the sex it was in a different their distinguished the sex chromosomes, and the rest of the chromosomes were they named as in a autosomes. So which they are functioning, there is that X the sex chromosomes we are named is called as what, that is in a completely in a autosomes. Okay, and Here, the grasshopper. The grasshopper is an example of the XO type of the sexual determination, which is in the males and having X chromosomes based on the autosomes, where the females has the pair of X chromosome. So for example, here taking as an X chromosome, and XY chromosome. So here is X is the completely expressed to as that character. They given clearly here. The determination of the sex chromosomal differences, capital A, small b. The both the humans, the Drosophila, the females they pairs as an XX chromosomes and homogenetic they form, and the male XY the heterozygous the composition. Many birds, females, as a pair of the dissimilar chromosomes, they show as what in a completely ZZ, that male in a ZW, that is what it is in a completely female characters. Okay, next. After this observational to lead that in an investigation the member of the species to understand the mechanism of the sex determination. So these all observations based on, then only we completely they explain about as in the sex determination mechanism. Sex determination mechanism. Okay, and here in number of other insects, mammals, including as an XY type sex determination, seen that in both male and female the same number of chromosomes. So in the some insects we have in what XY chromosome and XX chromosomes also we are noticed. Among the male, X chromosome is present, but in counterpart of the distinctly the smaller the called as Y chromosome. In that smaller one is also there present in that males, that is in a Y chromosome, and female they have in a pair of X chromosome. The both male and female bears on the same chromosome, that is called as what completely autosome. So when they are completely present as in one X chromosome and Y chromosome, this is called as what, that is in autosomes. And here's the male's autosomes plus XY, it is in a completely XY, and there while the female the autosomes in XS, so XS plus, it is in a XS plus. Next, the human beings and the Drosophila, the males has an X chromosome, the Y chromosomes, where the females having a pair of X chromosomes. They having as in a pair of X chromosomes present as in the autosomes in that in a females. In the females. Okay. Next, after whereas the females here, whereas the females the X chromosome in the autosomes complete, yeah. In that above the description of you have in the study about as two types of the sex determining mechanism. So when you observe as two types of sex determination mechanism, one is in a XO type and the one is in a XY type. One is in a XY type. The both in male produce the two different types of gametes. So they are produced as in both completely male gametes. Here either with or without X chromosome, or some gametes are X chromosome in some without in a Y chromosome. So sometimes they get produced as in a O and sometimes they could get produced as in a Y chromosome. Such types of the sex determination mechanism, it is in a designed to be explained as in a male heterogamy. It shows as in a male heterogamy means it is show as in two different characters, right? That is. And in some organisms, example in birds and different mechanisms sex determination to observe in this case of the total number of chromosomes on the same both males and females. So the total number of chromosomes as present as both and male and female. Here for example they have 23, also it comes as in completely the 23 pairs of the chromosomes as forms. But two different types of the gametes, the term sex chromosomes, that is in a sex chromosomes the produce as in a females. They get completely sex chromosome produced as which one? That only for the female. So that is called as what? The female heterogamy. So it is a complete the females to get produced in two different kinds of the chromosomes. Next, in orderly having a discussion with as in a mechanism of sex determination, the described earlier. So it is completely explained about as in that earlier only. The two different sex chromosomes, so the female birds has been as in a differently as in a Z and W. So the having as in a ZW is present, and the organisms the females to have in a ZW. Here the other organisms we see as in a ZW that has in the same female sex organs as present, whereas in a males having the pair of the ZY chromosome. So they have only for that in a Z is what? It is a part of the in a male, it shows as in a autosome. It is in a completely autosome. Remaining to discuss in next class. Thank you for watching. Like, share and subscribe.